Hi, my name is Brian Kim. I'm an assistant professor in electrical and computer engineering department. And today I'll talk about the development of super-resolution neurochemical-based neurointerface, which is a part of the CECS-COM pilot award that's funded in 2021. Just briefly to talk about myself, as I said, I'm a faculty in electrical and computer engineering. My main research is two different things. One is a high-density neurochemical sensor, and the second project is point-of-care diagnostics. So today we'll talk about the first part. My sources of funding for supporting my research is NIH, NSF, and DoD. So today we'll talk about the neurochemical-based neurointerface, but before we get there, we'll talk some motivations here. So I'm sure many of you are very familiar with the topic of neurointerface, especially with Elon Musk starting a Neuralink, and it's becoming a very well-known fact for general publics as well. So when we look at neurointerfaces, they serve multiple purposes, but essentially they help us understand the computation of brain. They can help us create better neuroprosthetics. That's shown in the middle here. And this one is the visual cortex where we're reading and being able to see that as well. And in order to create neurointerfaces, a number of different technology has been developed so far, where we can either have a direct implant in the brain that can measure neural spikes or local field potential, or we can measure from the surface of the skin, where we are measuring EEG, which is relatively smaller and slower, and has perhaps an indirect information of the brain intentions. And also you can measure directly from the surface rather than penetrating, which is called ECOG. And all these technologies have different advantages. Some ones are better in spatial resolution, some ones are better in temporal resolutions, and so on. But so far, penetrating electrode, which is shown on this side, has shown the best both temporal and spatial resolution. So one of the things that's going on in the field, within the field of penetrating electrode, is to increase the number of electrodes, because that could help us capture higher density of neural interfaces from the brain. However, one thing that people are currently realizing is the number of electrodes. We can just increase the sheer number, but that's not everything. And this is one on the left side that shows really high array of EEG, which is fantastic. And also this one is the electrode array that's developed by Elon Musk Neuralink that has 1,000 electrodes, which is really, really fascinating. One of the problems with increasing the number of electrodes is what I call crosstalk. So if you see an example here where you have a surface electrode versus a penetrating electrode here, if neuron spike fires, you will see the action potential or local field potential from multiple electrodes that's adjacent. So that what that means is if you have a lot more electrodes in a given space, there is an actual effective limit at some point where all you're increasing is number of electrodes, and they're not really resolving single neurons. So penetrating electrode really solved a lot of this issue. And one way they're solving this issue is by penetrating the tissue directly. And therefore you're isolating or insulating, so to say, and establishing a connection between one neuron to one electrode. And that way you can really improve the scalability of this technology to achieve better neural interfaces. One of the interests that I have is to kind of deviate away from those typical what we call electrophysiology methods to neurochemical sensing methods. So as everyone here who've taken biology class in high school are aware of, neurons release neurotransmitters. It could be dopamine, norepinephrine, acetylcholine, glutamate, you name it. They release neurotransmitters. And these are usually happening at the synapses, and they are highly correlated with their activities. If there's neural impulses, they release neurotransmitter at their synaptic cleft. And we have ways to measure these directly from electrodes. Now, the reason I'm interested in looking at neurochemical is because I believe that there is stronger potential in creating a more scalable technology than relying on typical electrophysiology that is prone to crosstalk, where you're limited to a certain number of electrodes, and improving the number of electrodes doesn't necessarily improve your scalability. And it has something to do with the diffusion characteristics of these neurotransmitters. So if there's a site where they're released, they go through a diffusion and they reach number of adjacent electrodes. So what we are currently studying is to figure out a way to use this diffusion characteristics to improve the spatial resolution of neurochemical imaging. So for example, here, if you have 4 electrodes, right, we can always create more electrodes. So we'll talk about that in a minute. But let's say you have 4 electrodes. In general term, right, if you're doing normal electrophysiology or neurochemical recording, that means you have 4 points in your pixel of neurochemical imaging, typically, right? But if you look at the diffusion characteristic, if you have, let's say, this is the release site, they would diffuse and they will get collected from multiple electrodes, right? Of course, one vesicle that's being released through exocytosis releases tens of thousands, sometimes millions of neurotransmitters. So all the electrodes will measure them. But what we know also is based on the diffusion characteristic, they will be measuring different amount, right? Some of them that's closer will measure higher amount. Some of them that's farther will measure less amount. But this also depends on the geometry that they're traveling as well. And we know that we can use random walk simulation to accurately characterize the diffusion characteristic as long as we know the geometry. And if we do that, we can look at this recording and figure out and reverse calculate where it might actually have come from, right? So neurotransmitter release is so small that we can't see it. And when you have 4 electrodes, your spatial resolution is very limited. But with the diffusion characteristic and us using the random walk simulation, we can do a localization of that position very precisely. So that's what we've been working on. And this project only started a few months ago, so we are still at the stage of collecting data. But in that regards, one of the other things that I wanted to show is the high density neurochemical probe. So this technology improves the spatial resolution, but you still need a large high density neurochemical sensor that can capture release from really large area of brain tissue. So we have in the past developed a CMOS chip, which is basically like a computer chip that integrates 1024 neurochemical sensors in a really packed space. So you can see that it's roughly about 1 millimeter and 1 millimeter, and I won't get into the circuit side of all these things, but each individual squares that you're seeing here that's really tiny corresponds to 1 neurochemical amplifier and 1 neurochemical electrode that's actually on the surface. So with that, we've tested the performance of this device, and it matches high quality electrophysiology amplifier that you can get that usually cost around $30,000 to $50,000. Keep in mind, $30,000 to $50,000 conventional electrophysiology amplifier has 1 channel of neurochemical sensing capability. This chip has 1,000, and we can usually fabricate under about $100. So this is very cost effective as well. Mostly we've been working on in vitro system where this chip is integrated in a microfluidic device and the cells are directly cultured on top of it. And my collaborator here, Dr. Sugaya, is helping us with the cell culture. And you can see this is the package device where you have the surface where 1,000 electrodes is exposed for our application. We are currently also working on in vivo application. And it's actually very simple. Since we have already the circuit, the only thing that we have to do is to change the form factor. And this is going to be fabricated later this month. So we are really excited to get this chip back sometime soon, hopefully early next year so that we can get tested. And this will be, in a way, the first high density neurochemical sensor that will be tested in vivo anywhere. So we are really excited about that research opportunity. The nice thing about our system in general is that we have the highest neurochemical channel count reported to date. There is no competitor that's anywhere close to where we are able to do. The level of noise level for each neurochemical sensor that we are achieving is, as I said, comparable to the high quality patch clamp amplifier. That's what we usually call it. And we have also shown that you can use this setup and culture directly the cells directly on top of it and have single vesicle resolution measurement. So usually we don't measure really broad, diffuse characteristics of neurotransmitter, but we can usually resolve down to single vesicle release, which is about 40 to 50 nanometers in size. This shows some of our early studies. So this one on the left side is the data that we're showing where we're using our sensor for neurochemical imaging. The one on the left is hydrogen peroxide diffusing above our surface that we are measuring. So it started with light blue where it means you don't have any oxidative current or neurochemical signal. And as it goes into higher time frame and the time passes, you're seeing more purple, which corresponds to higher level of oxidation current due to neurotransmitter diffusing, or hydrogen peroxide in this case that's diffusing. And the one on the right is the same type of measurement for dopamine specifically. As far as the super resolution reconstruction is going, we are early in the stage of developing the algorithm. So basically the way it's going to work is we're going to use our current hardware platform, and that's not going to change. But we'll have this algorithm that we'll build that will do the localization using the random walk simulation, and we'll be able to create it. And our early data suggests down here, this is corresponding to the measurement from the forward electrode, where the release site was changing. So we intentionally changed it and see if you can actually resolve it, right? So we're hoping that we can resolve down to a few hundreds of nanometer. Therefore, we can create a system that has about 100 nanometer or 500 nanometer spatial resolution, which would be incredible. So this one is showing the fact that we are changing the X location by 500 nanometer, and you can see a huge difference in signal, and that therefore we will be able to identify them. And secondly, here you're changing the Z location of the release site. So we haven't really talked a lot about 3D space, but we are actually considering 3D space, although the presentation appears to be 2D in space. And as we are changing the Z location of the release site in micron, we can also resolve that, not by the ratio of signal strength on each electrode, but on the delay or the duration of the spike in this case, which I call it half-width, 27 milliseconds and 31 milliseconds. So with that, I would like to thank you for paying attention to my presentation, and I appreciate for the CCX.com Pilot Award that enabled this research, and I'll take any of your questions.